Hello, my name is David Winogradoff. I'll be speaking on behalf of my advisor, Professor Aksimentiev, myself, and everyone who worked on this project. Thank you for the opportunity to talk today at this symposium. I'll be talking about the molecular mechanism of selective transport across the nuclear pore complex. This is an ongoing project, and today I'll show what we've done, the progress we've made in the past year. So what distinguishes eukaryotes from simpler life is the nucleus itself, and in addition to providing a place for storage and protection of the DNA, it also provides a layer of regulation for the expression of that DNA. And one of those layers is the nuclear pore complex itself. So these pores that you see here are the only way that RNA and proteins are able to cross the nuclear envelope. And so one thing that's central to this passage is this central mesh. So NUP is the abbreviation of nucleoporin, which is just the term for a protein in the nuclear pore. And one of the reasons that this is difficult to study both in experiment and in simulation is that these NUPs, called FG NUPs, are intrinsically disordered, which makes it difficult to study. So there's been many proposed theories for how transport happens, but really we don't know yet. The reason we say selective is because larger molecules can only pass through when they're accompanied by a transport factor. So, all right. So because this is such a large system and it's difficult to study in vivo, this is literally thousands of proteins, the Decker lab, The Decker lab from TU Delft that we're collaborating with, and also the Chait lab a few years earlier, found a way to synthetically mimic the properties of the nuclear pore. So what they did was to drill a hole of various size in a solid state membrane of their design and to coat that with NUPs. And they did this with several different species. And first what they did was to just apply an electric field and to calculate the conductance. And then also they were able to drive a protein through and to measure the current during that process. So this is beneficial in the sense that it gives us control over the geometry of the pore itself. And also because we're applying an electric field, we can examine the electrical properties. So we set out to do this same thing in MD simulations, since we can also apply an electric field, and we have the added benefit of knowing the position of all the different atoms throughout the simulation. So what we did was we applied forces, half harmonic potentials, to just the protein, fixing it in this central region, equilibrated it, and then applied a voltage across it. Now we chose NSP1, which stands for non-structured protein 1, since this is one of the important NUPs in the central channel of the human nuclear pore complex. This is about 600 residues long, so what we decided to do was to allow this protein to explore more conformational space, we divided it into fragments. And to do a similar process to what was done in experiment, we varied the size of the cross-sectional area, keeping the Z the same, and in this way, we varied the protein content within that central region. And I'll show you the setup of one particular system. So starting with the protein, you then add water and ions. Now if you have good eyes, you can track this one particular potassium ion that I highlighted. You apply an electric field, and you can see that the motion of this ion is somewhat stochastic in Brownian motion, but it has a preferred direction that goes along with the electric field. And what we can do is we can trace the motion of all these different ions, and from that we can calculate the current and also the conductivity, since we know the geometry. And this is just the formula. Essentially what you're doing is you're measuring instantaneously, you're measuring how much charge is passing some length along the z. So because we have periodic boundary conditions, you don't actually measure along the whole length of the system. And from here, we can, also knowing the geometry, we can calculate the conductivity, and we did this for all the different systems. And what we see is we see this transition, just as seen in an experiment, from conducting to not conducting. So at about 60% of protein volume, it switches to not being able to conduct any ions at all. So we were encouraged by this result in that we were able to match qualitatively the, what was seen in an experiment. And what we decided to do was to try to build an all-atom model of the entire nuclear pore complex. Now, this really only became possible about a year ago when the structure of about 80% of the nuclear pore complex, the ordered part, was mapped. And this was highlighted on the cover of Science about a year ago by the Holtz lab from Caltech. And what you see this nice symmetry. There's two outer rings, one for the cytoplasm, one for the nucleus, and there's this inner ring. Now, what they're missing, the 20% that's left over, is that intrinsically disordered part. And the way we approached modeling this was by using both coarse grain and then switching to all atom. The reason this is required is because the amount of conformational space that these proteins need to explore is very large, and it's something that we wouldn't be able to reach that time scale for a system of this size in all atom. So we chose the coarse grain potential that we chose is from the Onc lab. And the reason we chose that is because it was specifically designed for these nucleoporins. So they have a huge, so the red is simulation and the black is experiment. As you can see, these are about, and what they tuned against was Stokes radius, which is one measure of the size of the protein. And they plotted it on the x-axis with a charge to hydrophobic ratio. And you can see that there's quite a few NUPs that are much more hydrophobic than they are charged. And this is the data set from 2010. So what we did, so the reason I call them core NUPs is because the ordered part I consider to be the core, and then the NUPs that form the central mesh are actually the end-terminal tails, long end-terminal tails of these ordered NUPs. And so we took what they mapped in all atom and built a potential. And then in addition to just what was seen in all atom, we added a curved surface for the nuclear envelope itself. Now, so this is one of the simulations that we ran at coarse grain resolution. And what I'll do is to show the RG, the radius of gyration of the entire network as a function of time. So you can see that throughout this simulation, it's decreasing, and then we have one period where it goes up. And the reason for that is because we specifically changed their potential to have weaker bonds so that any tangling, any extreme tangling that would happen in this network would be able to be released. As you can see, there's, there are these channels that form, if you remember what it looks like at the end. And this is something that we need to investigate further and see if this is something that happens always or if it depends on the initial conformation. So one of the ways we analyze this simulation is to look at, we map the density of all these proteins throughout the entire coarse grain simulation. And there's a total of about 50,000 residues. We see it covers most of the space, although there's some parts you can see that are not completely covered. About 25% of all the residues are hydrophobic, and you see it forms the backbone of some kind of network. And from that starting point, we were able to map that coarse grain final conformation to all atom, shown in purple. Then we added the nuclear envelope, so this is just a curved lipid bilayer in a toroidal shape. And we added water and ions to the whole system. So before running anything on blue waters of this entire system, first we calculated an estimate of what the conductance would be given a certain applied voltage. And this is a protocol that was developed by Aleksi and my advisor, Professor Aksimentiev. And basically the way it works is that you split the protein into slices vertically, and then for each of those vertical slices you determine what the resistance would be, and then you can add those resistances together. So this middle line would be the resistance of just one voxel, and then that formula there was for an entire slice, and you add up the resistances of all the slices. And applying 100 millivolt voltage, we see that from this protocol we get about 65 nanosiemens, and from experiment we also developed an estimate, and we see that it's a little bit lower, but it's relatively close to what we predicted from this protocol. Now, ultimately, what we've been able to do just recently is to build the entire thing and run it on blue waters. It's about 140 million atoms in total. This is starting to push the limits of what's possible with an AMD, although in theory you can get quite a bit larger. So, and we've performed benchmark simulations, and we still need to increase the node count and to see what kind of timing we would get there. So we see that even with 200 nodes, which is quite large for a small system, we only get about half a nanosecond per day. So we'll perform more benchmarks to see what's the optimal number of nodes to run this on. So in conclusion, we saw that in all atom simulations, we were able to match the electrical properties seen in experiment for a network of NUPs. We built the first complete all-atom model of a nuclear pore complex, which in total was about 140 million atoms. We've started to characterize the electrical properties of that entire system, and we'll also continue to do that with simulations where we apply an electric field. And lastly, as a future direction, we'll model the diffusion process of proteins and RNA across the nuclear pore. And we'll do this both in coarse grain and all-atom resolution. So these are the people that worked on this project. My advisor, Aleksi. Chris Maffeo is a developer that is the current developer of the engine for the coarse grain simulations. Max is our host today, and he helped get the project started, get it off the ground. Chen Yu helped with the lipid bilayer. Aleksi I didn't have a picture of, though he helped quite a bit. Kay Stecker is our collaborator, and the potential for the coarse grain simulation was actually provided by the Enclave. So we thank them all for that, and Blue Waters and Illinois for the computational time to do this project. Thank you very much.
We have 5 minutes for questions